Let's look at some animal cells and plant cells. So here's an animal cell. We're going to have all of the parts that we kind of talked about earlier. We've got our peroxisomes, which will neutralize peroxides. We got our plasma membrane, our rough ER, rough endoplasmic reticulum, which is beaded with ribosomes, so kind of the construction site for protein synthesis, ribosomes being the construction workers for protein synthesis. We got our nucleus, which has a nuclear envelope, bilayered nuclear envelope. We've got our nucleolus within the nucleus. We've got our mitochondrion. This is an electron microscope scan of it. Here's a nucleus, and here's the nucleolus. And then in our rough endoplasmic reticulum, all these little dots, those are going to be the ribosomes. So here, this is just showing what a smooth endoplasmic reticulum would look like. If you remember from cell replication or mitosis, meiosis, we talked a little bit about centrioles. Those are going to be the organelles that create those spindle fibers made out of microtubules that kind of orchestrate the process of nuclear division. And then here's just showing, outside of the cell, inside of the cell. Now let's look at a plant cell. Again, your rough endoplasmic reticulum is going to look pretty similar to a plant cell rough endoplasmic reticulum. Same with the smooth endoplasmic reticulum. The Golgi, remember, that's going to be the post office in a sense, and then you've got some free ribosomes over here. You've got the vacuole, the central vacuole in this plant cell. Not going to typically find that in many animal cells, or at least as large as what you would find in an animal cell or plant cell. Plant cells, you'll typically find chloroplasts, which are going to be those organelles that photosynthesize stuff. And then here, peroxisome again, that's going to be the organelle that neutralizes peroxides. On plant cells, you will oftentimes find a cell wall made out of cellulose. And then here is an electron microscope scan of a chloroplast. All right, so the main treat of this video is the cytoskeleton. Let's talk about the cytoskeleton. So the cytoskeleton is going to maintain, it's going to help to maintain your cell shape and support that cell shape. It's going to provide mechanisms for cell movement, so things like your flagella or your cilia. And then it's going to also act as kind of a track for motor proteins, which helps move materials within different locations of the cell, or two different locations of the cell. So there's three different types of cytoskeletal components, which we're going to talk about in detail. We've got microfilaments, intermediate filaments, and microtubules. So to start off with microfilaments, those are going to be made of the protein actin. So actin exists as single filaments in bundles or in networks. So you've got F-actin, which is filamentous, polymer, meaning it's grouped together, and then you have G-actin, which is globular, typically when we're dealing with a monomer. Microfilaments are needed for cell contraction. They help maintain cell shape, so you're going to find them often on the edge of your cell membrane, just inside of your cell membrane. And they're going to be involved heavily in cytoplasmic streaming and the formation of pseudopodia. So first I'm going to show you a video of an amoeba. So here's a video of an amoeba moving. So each of those little lobes that are developing are called pseudopodia, or fake feet is what it roughly translates to. And the way it's moving is through cytoplasmic streaming. All orchestrated by actin, the protein actin. Next, we have a video of a neuron undergoing cytoplasmic streaming. So here's a neuron moving in a determined fashion through the use of actin. Next, we're going to look at a video that depicts treadmilling. So here's an abstract depiction of treadmilling. You're going to be dealing with G-actin as a monomer. You can have a dimer, trimer, a polychain of G-actin. You're going to have two different forms. The darker is bound to ATP, and the lighter is bound to ADP. And then the hand is just depicting the exchange protein, the thing that swaps it from ATP to ADP and vice versa. So you have ADP, which is swapped to ATP, which then has an affinity for the darker region, which is the other actin with ATP bound. So it's an energy-intensive process. So you take the actin from the back and change its conformation, and then it binds to the actin in the front. So that was microfilaments. Those are going to be the smallest of all of the cytoskeleton components. Next, we have intermediate filaments. These are going to find only in multicellular organisms, and what they do is they form these rope-like structures. They have a tendency to resist tension, and they're the main anchor point for your cells. They maintain the positions of all of your organelles. So they all kind of stay in place because of your intermediate filaments. Next, we have microtubules, which are pretty interesting. So they're hollow cylinders made from a protein called tubulin, which exists in dimer subunits. Di meaning two, so it's a dimer subunit. So many of those together, many dimers, or many of the tubulin dimers together will make you could say a strand or a microtubule. So microtubules are going to be used for their rigid structure. And importantly, they serve as motor protein tracks. So they're kind of like the highway system in some cells. Typically, when you send things from one area of a cell to another area of a cell in a vesicle, it's going to walk along a motor protein track or down a microtubule. And so they're going to be heavily involved in neuronal transport, which I'll show you in a little bit. So common uses of microtubules, or common microtubules we'll see within cells, are flagella. Those are going to be those long tail-like structures that provide movement by rotating and whipping, which pushes and pulls the cell through its environment. Next, we have cilia. Typically, when you have cilia, you have more than one. Those are going to be those short hair-like structures that will provide locomotion by moving fluid over the cell. So if you've seen paramecium under the microscope, they've got many cilia that allows them to move through solution. And then next, we're going to talk about axons. Axons are going to be these large appendages, or these large tubes, you could say, that connect the cell body to the axon terminal in a traditional neuron. So first, let's talk about cilia a little bit. Cilia, the way they move is going to be kind of conducted by the interaction between the microtubule, dynein, and nexin. So dynein is going to be a protein that actually allows the microtubules to slide back and forth along each other. So here you have a negative end and a positive end, and what it's going to do is it's going to move the microtubules in this direction, so in opposite directions of one another. So the dynein shape will change through the use of ATP. So it's actually an energetic process. It's an active process to whip around cilia, for example. And it's going to move the microtubules, therefore bending the cilia. And then next we have nexin, which are kind of like the anchors between your microtubules. Those are going to be, or nexin are going to be proteins that attach microtubules together. So here we show that dynein, that's going to be that protein that will change in conformation, allowing the movement of your microtubules in opposite directions of one another. So if you had a bunch of dynein in between two microtubules, what's going to happen is they will indefinitely move in opposite directions until they slide off of one another. However, as I mentioned, we have these little proteins called nexin, and that's going to provide an anchor or an attachment site between your microtubules, so that when your dynein changes in conformation with the use of energy, it actually, instead of causing these microtubules to slide indefinitely along each other, what happens is they actually just bend. So it's the nexin combined with the dynein and the between the microtubules that allows them to bend rather than just sliding off of one another. Again, you're going to find lots of cilia located on the surface of paramecium, and that's what allows for them to move. Okay, so let's say we're not dealing with cilia, and we're just trying to transport proteins from one location to another. Well, now we're dealing with motor protein tracks. So motor proteins will move along microtubules. Those microtubules kind of serve as like a highway. Now dynein, we just talked about dynein. Remember, that's that protein that will kind of move along a microtubule, and if it's sandwiched between two microtubules, it causes them to slide in opposite directions of one another. However, if it's not sandwiched between two microtubules, what it does is it just walks along the microtubule. So we also have this other protein called kinesin. So kinesin is going to carry vesicles, or you could just say cargo, towards the positive end of a microtubule, whereas dynein is going to transport cargo or carry materials to the negative end of a microtubule. So like I mentioned earlier, axons have this, or neurons will have this part of the cell called the axon. And typically, you're going to have cell signal being received over near the dendrites, typically located around the cell body of a neuron. So here's the cell body. And then you have this axon, this long axon. And at the very end of this axon, you have an axon terminal. Now, in order to get stuff from your cell body, which is going to house your nucleus and your rough endoplasmic reticulum and have a lot of ribosomes over there, you're going to synthesize a lot of proteins over there. If you want to get those products out to the axon terminal, well, you're going to need to send those vesicles down the axon to get to it. And so this is where your transport proteins are going to come into play. So peptides are synthesized in the rough ER, packaged by the Golgi apparatus, put into a vesicle, and then it's sent through fast, or you could just say axonal transport, is going to walk the vesicles down those microtubules to the axon terminal. So it's not only vesicles that are going to be moving. Sometimes it's going to be organelles or things like mitochondria that will move down those axon terminals. And then once you get to the very end, if you're a neuron, you might store those vesicles containing neurotransmitter, for example. And if the cell receives signal to release those neurotransmitter off to a neighboring cell or off to a neighboring neuron, for example, those vesicles will fuse with the axon button or the axon terminal and then release those neurotransmitter or cell signals into the extracellular space. Now you might create new vesicles down here or just simply recycle vesicles, send them back down. We would call this retrograde axonal transport. And they could serve whatever function back towards the cell body or just get degraded. So if you fuse them with a lysosome, they could then be degraded and recycled, the cell components or the vesicle components. All right. So next we have the cell wall. So that's going to typically be composed of cellulose fibers. So cellulose, we know that's going to be a complex carbohydrate. And that's going to be embedded in a matrix of other complex polysaccharides and proteins. Now, you're not going to find cell walls in animal cells at all whatsoever. Your cell walls are going to provide rigid structure, and they serve as a barrier to larger organisms like herbivores that may cause plant damage, you could say, damage, disease, infection. So they're kind of a protective structure. Next, we have extracellular matrix. Your extracellular matrix is generally going to be created by the cells around it. So the cells around an extracellular matrix, extracellular just means outside of the cell. It's going to, your surrounding cells will secrete all of the molecules or components of that extracellular matrix. So Your extracellular matrix is going to be composed of fibrous proteins, things like collagen, which is a complex protein, or glycoproteins, or linker proteins. You're going to find extracellular matrix in your bone. You're going to find extracellular matrix in your skin. Some of the functions of extracellular matrix is that it holds together tissues, various tissues together. It's going to help filter material passing between tissues. It's also going to kind of house a lot of your immune cells. It's going to help orient cell movements, and then importantly, it's going to play a role in chemical signaling. It's kind of a vector for signaling between cells. Lastly, we're going to determine what all cells have. So all cells are going to have some kind of genetic material. So in this case, it's labeling the chromosome of a prokaryote. You've got chromosomes, or you could say your genetic material in chromatin form, your chromosomes in chromatin form in the nuclei here. You're also going to have ribosomes, both in prokaryotes and animal cells and plant cells. You're going to have cytoplasm in all of them, so that juice within that cell, that pulpy juice, you're going to find that in prokaryotes and eukaryotes, including animal cells, plant cells. All of these cells are going to have a plasma membrane. So all of them have a plasma membrane. Oftentimes people think, well, because plant cells typically have cell walls, they just have a cell wall, and I think they assume that the cell wall replaces the cell membrane. Well, the cell wall is actually going to be just outside of the cell membrane. So your plant cells have cell wall as well as a cell membrane. Now you might find a cell wall in your prokaryotic cells. You might find cell wall in your plant cells. However, you're not going to find cell wall in your animal cells. Okay. So what do we have? Cytoplasm, genetic material, ribosomes, and cell membrane. Those are the four things that all cells will have.